Hi, Dan Guerra here from VerEvMed. Today we're going to continue on our discussion of reactive oxygen in context with the larger discussion we're having on human aging. So let's get started. Specifically, let's talk about some biological sources of reactive oxygen species, which we're calling ROS. Now, we mentioned last time, but in case you didn't watch that or don't know where to find it, let me recapitulate that reactive oxygen is a very common product of metabolism in the cell, particularly in all aerobic organisms. So we have something called the electron transport chain, which is in the inner membrane of the mitochondria. This process of transporting electrons is associated with pumping protons across that membrane into the intramembrane space between the inner and outer membrane of the mitochondrion. And then when those protons are pumped back in through a proton pumping ATPase, we make ATP. And then that process we're oxidizing NADH or FADH2, transporting electrons along a gradient, and ultimately those electrons along with the protons are going to take molecular oxygen and convert it to water. Okay, so here is the electron transport chain, the specific complexes in that inner mitochondrial membrane. They're broken up into complexes, and perhaps you remember this from a basic physiology course or a basic biochemistry course. So I'm just refreshing you rather quickly here. But you have complex 1 here, which is an NADH dehydrogenase. So that's important to know that dehydrogenases are involved in redox reactions. It has a flavin mononucleotide associated with it. It also has an iron sulfur center. And there's a secondary enzyme which basically rebuilds the redox state, and that's called NADH ubiquinone oxidoreductase. Complex 2, and that's going to drive its electrons into the ubiquinone pool, which is right here. Now, let's take a look at complex 2. Complex 2 enters not as NAD, but as FAD. So basically, it's coming from the succinate dehydrogenase reaction directly from the tricarboxylic acid cycle, that is Krebs cycle, also found in the mitochondria. So we're going to be starting at the oxidation state of FAD, which is going to be a lower yield of energy, so we're not going to make as much ADP this way. But we're going to have FAD bound directly to this enzyme, which is associated with this complex. We're going to have iron sulfur centers. We're also going to have a B-type heme. Now, the auxiliary enzyme for complex 2 is going to be the one that's going to, again, rebuild that redox state, and that's going to be a succinate ubiquinone oxidoreductase. Again, drive electrons into the ubiquinone pool, and then those electrons are going to make it to complex 3. At complex 3, we have a series of proteins, cytochrome BC1. There are 2 heme-type structures. There's a risky iron sulfur center that's named after the person who discovered it. There are also start to see C-type of cytochromes. In particular, here is cytochrome C1. The rebuilding enzyme that's associated with complex 3 is ubiquinol. Notice that's the alcohol, right? QH2, cytochrome C oxidoreductase. Okay, so it's going to rebuild the oxidation state of this pool so we can rebuild the oxidation state of these 2 pools of proteins embedded in the membrane so that we can allow for the introduction of NADH and FADH2 from the oxidation of things like fuel, such as carbohydrates, amino acids via transamination reactions, of course, and fatty acids. The electrons that are moving from complex 3 are going to move through a cytochrome C complex and ultimately to the terminal oxidase. This is complex 4 in the electron transport chain. It's going to have a unique set of cytochromes called AA3s. So it's also going to not have iron, it's going to have copper. So it's lower oxidation state here. So electrons are going to move from here to there according to the redox state of this system. Ultimately, the electron acceptor is going to be molecular oxygen, as shown here, and we're going to make water from it. Okay, so this is the final reduction of molecular oxygen to water, and that terminal oxidase is called cytochrome C oxidase. All right, so remember last time, well, again, I shouldn't suppose that you do, but last time we talked a little bit about inorganic chemistry, and we talked about the cell potential. So the positive, we have a positive E0, which is the cell potential. You have a spontaneous reaction. That means it's going to move forward. There's nothing, unless something is going to stop it. because of some blockage of electrons, we're going to move from a level of relatively negative E naughts to positive E naughts because the electrons are going to move down this cascade. So we're going to go from the couple, these are conjugate base pairs, okay, NAD, NADH. We're going to move to coenzyme Q. Then we're going to move the cytochromes, and ultimately we're going to move to oxygen. So we're going to move from a relatively negative E naught to a positive E naught. And because it's spontaneous at the positive level, we're going to go ahead and we're going to reduce that molecular oxygen to water. Okay. Here's shown again in more of an ultimatum type diagram, giving you specific measurements in volts, okay, not in joule coulomb, which is more common for biology, but 0.32 volts, moving through almost an electric state here at the coenzyme Q pool. Then we're starting to move more positive, the cytochrome C oxidoreductase, and ultimately at the cytochrome C terminal oxidase down to molecular oxygen. Okay, so a negative to a positive, the electrons are going to move in this direction. And along the way, remember, we're going to be making some partially reduced forms of molecular oxygen. So superoxide, hydroxyl radical compounds like that, hydrogen peroxide, and those, of course, are going to be dangerous to the cell. So when the mitochondrion is functioning in its normal state, you're going to make reactive oxygen, and because you make reactive oxygen, you can cause damage to the cell. That's the price we pay for using molecular oxygen as our ultimate electron acceptor. We make a lot more ATP that way as aerobes. So let's discuss this now, make a narrative of what we just looked at in figurative. Electron transport chain is initiated by the reaction of an intermediate in metabolic reactions. And of course, that's going to be nicotinamide adenine dinucleotide reduced form. You're going to make that, for example, in the Krebs cycle. The NADH is going to be made in the Krebs cycle because you're going to be oxidizing carbon coming into that Krebs cycle. You're also going to make NADH and indeed FADH2 from the oxidation of fatty acids, which happens during fasting. So anyways, the oxidation reaction is where two hydrogen atoms coming from, now we're talking NADH, H+, or that is two hydrogen ions and two electrons, are removed from the organic metabolite. The metabolites are usually from the citric acid cycles I just mentioned or fatty acids. So the reaction can be abstracted in this way, where M can be any metabolite. You got a reduced metabolite, you got NAD+, that's the oxidized form. You make reduced NAD, that's NADH. You've got a hydrogen here and a hydrogen there. Those are your two hydrogens, right? Now you have the oxidized product, right, like the organic compound, and you're going to get energy from this reaction, which is ultimately going to be driven by that proton motive force in the intermitochondrial membrane to make ATP. You make actually 3 ATP per mole of NADH. Okay. All right. So one hydrogen is removed with 2 electrons. We call that a hydride ion, while the other is removed just as a proton or a positive ion. Usually the metabolite is some sort of alcohol, and we're oxidizing to a ketone. That's a typical organic reaction, alcohol to a ketone. Purpose of the other 7 steps in the entire electron transport chain are just to rebuild, right? So you're going to pass along those protons and electrons to eventually react with oxygen. You're going to reduce oxygen to water. You're going to conserve energy along the way. It's not going to be perfectly conserved, of course, because you're dealing with biological systems. So you're going to make 3 ATPs, though, which is a really good amount, considering the oxidation you're doing. And you're going to regenerate all those coenzymes so that you can go back to being oxidizing agents and carry on and facilitate the ETC, that's electron transport chain. Now remember, this is again going more abstract, back to more or less straightforward chemistry. How do we transfer electrons? We do it in like one of 4 mechanisms. There's maybe a 5th one, but right now let's just talk about the 4. You have a direct electron transfer, as when we're talking about the galvanic cells sort of moving from iron to copper, remember? So for example, Fe2 plus copper 2 goes to copper plus Fe3, right? So what have we done here? We've oxidized the Fe2 and we've reduced the copper here, right? Okay. That happens in those iron sulfur centers that we just talked about in electron transport chain. So that's there. Second, as a hydrogen atom. Again, a reduced form of some organic metabolite, oxidized. You strip off those electrons and protons. And that AH2, of course, is going to be in the A. That's going to be your redox couple or redox pair, right? And they're going to possess a tendency to reduce this subsequent compound in the chain, right? Just like we saw going from NADH oxido reductase to the ubiquinone pool, the cytochrome C, etc. So that's what we're talking about here. This could be like NADH. This could be the oxidized ubiquinone. You make NAD, and now you make the reduced ubiquinone, for example. You can also move as a hydride, which we talked about before. That is hydrogen with 2 paired electrons. So again, you've got the reduced substrate. You react it with a proton, and now you've made the oxidized product. You've got the hydride and the proton. Okay, so that's where you're at with that. This proton can come from various sources. The final way to transfer electrons is direct combination with molecular oxygen. And notice these other ones don't require oxygen, even though we call them oxidations. Right? Oxidation simply means removing electrons. But when you react with oxygen, we can call these things oxygenases, and you can directly react a compound, a reduced form of an organic compound like a fatty acid, and react it with molecular oxygen. And you could either make an alcohol, or you can go ahead and make a completely oxygenated product, okay, where both atoms of molecular oxygen are introduced. And this is, for example, what happens in prostaglandin biosynthesis, the products of cyclooxygenase, lipoxygenase, and P450 oxygenases, okay, which we'll talk about later, probably. So that was an abstraction. Now let's talk about the kinds of enzymes involved here. 4 types. We're really going to concentrate on oxidases and dehydrogenases here. Later on, we'll talk about hydroperoxidases and oxygenases. Oxidases do use oxygen as an electron acceptor. Dehydrogenases, as we looked at just recently in the electron transport chain, they're going to be catalyzing a transfer of hydrogen and electrons. So you're going to go from a donor, which is going to get oxidized, to an acceptor, which is going to get reduced, okay? We also have those hydroperoxidases, and there we're talking about using hydrogen peroxide as the oxidizing agent. And then we have the oxygenases, which I just mentioned, is directly utilizing diatomic molecular oxygen. So oxidases and dehydrogenases are involved in respiration. That's what we call coupling oxidative reactions of the electron transport chain with the phosphorylation of ADP to PI. We call that oxidative phosphorylation. That's also known as aerobic respiration, mitochondrial aerobic respiration. Now, the hydroperoxidases neutralize, one of the things they do in the cell is neutralize free radicals. So those are good agents. Those are good players, usually. And oxygenases often also are good players. They remove xenobiotics, okay? So by oxygenating something, you can remove via biotransformation some xenobiotics, of which many are taken in the diet and from the environment. So oxidases catalyze the removal of hydrogen from a substrate with the involvement of oxygen as the hydrogen acceptor. 2 forms of oxidases. Some of them are copper-containing, such as the cytochrome oxidase. That's the terminal oxidase in the electron transport chain. And others are flavoproteins. That means they have usually a flavin, such as flavin adenine dinucleotide or flavin mononucleotide. Okay, those are organic compounds that are involved as electron acceptors. Common among those are L-amino acid oxidases and xanthine oxidases. So the metabolism of amino acids, the oxidation of amino acids, and also in nucleotide oxidation, is purine nucleotide oxidation. So what happens here, you've got the reduced substrate, you've got molecular oxygen here, you're only using 1/2 of the oxygen, the diatomic, you make water, right, and you form the oxidized product. Also, you can include the direct, complete oxygen to make hydrogen peroxide, right, shown here. We looked at this actually last time. All right, here are dehydrogenases. 2 main functions. They transfer hydrogen from one substrate to another in a coupled redox reaction, and they are the big players in the electron transport chain. Dehydrogenases use coenzymes. Here we're talking about the nicotinamides and flavins. Of course, as we've been mentioning, those are going to be hydrogen carriers, right? Now, one of them also, recall, was ubiquinol. Now, ubiquinol is a lipid, and it's going to accept electrons as well. It's embedded in the intermitochondrial membrane. Remember, ubiquinol is made from mevalonic acid, so it's a prenyl lipid. It's one of those lipids that may become deficient. It's called Q10 in popular jargon, can become deficient with high chronic use of statins. Right? At least there's some implication of that, because you inhibit the early stages of the biosynthesis of those prenyl lipids, and ubiquinol is going to be one of those earlier products in prenyl lipid synthesis. All right. So again, you start off with reduced substrate, and you're going to oxidize it. In so doing, you have to then, because you're carrying out oxidation, you have to reduce a carrier molecule. In order to be able to reoxidize that carrier molecule, you're going to have another compound which will oxidize it and go from the reduced state to oxidized state. And then ultimately, you're going to have electrons here. It's going to pass those electrons off. Ultimately, in electron transport chain, it's going to be passing them off to molecular oxygen. Now, let's talk about something very specific biological, and we'll be very brief today. Now, there is a website that's called Gene Cards. So if you have a particular gene or protein and you want to know something about it, you can go to this website. It's free. And there is that particular one for the one we're talking about right now. But any gene you can plug in, and you can be able to show this up even on a Google search. So here I wanted to look up the aldehyde dehydrogenase 3A1 isoform of that gene, okay, of that gene product too. So this is typically not just an aldehyde dehydrogenase, but a fatty aldehyde dehydrogenase, which means the substrate is going to be a fatty aldehyde, long chain hydrocarbon, usually C6, C9, C15 even. Now, that fatty aldehyde dehydrogenase, or just the aldehyde dehydrogenase, will oxidize all kinds of aldehydes. Principal among them are some long chain fatty aldehydes. And you're going to make an acid. Okay, so aldehyde to an acid, it's oxidation. So they're involved in the detoxification of alcohol-derived acid aldehyde. So when people ingest ethanol, alcohol dehydrogenase makes acid aldehyde. This is a very cytotoxic compound. So we have an enzyme which takes acid aldehyde and generates acetate, or acetic acid. That's an aldehyde dehydrogenase. In fact, this enzyme will do that reaction. But besides that, you also get from this enzyme, and enzymes in the family of ALDHs, metabolism of corticosteroids, biogenic amines, such as using tyrosine and tryptophan, neurotransmitters, the product of those reactions, such as the catecholamines, and lipid peroxidation. All of this is going to be utilizing these aldehyde dehydrogenase enzymes, okay? So obviously these are very important aspects in biochemical processes in the cell. The enzyme is encoded by, it's made as a cytoplasmic homodimer, and it preferentially oxidizes aromatic, that means it has ring structure, or usually medium chain, like I said, 6 carbon or more, saturated or even unsaturated aldehyde substrates. It promotes resistance. Now, this is why we're talking about it here, so it's really kind of cool. Resistance to ultraviolet light that we receive on Earth when we're, for example, out there getting the sun. One of the products that's formed during UV light interaction with the epidermis in the skin is the production of a compound known as 4-hydroxy-2-nonenal. This is generated by UV light-induced reactive oxygen causing the production of that aldehyde, that 4-hydroxy-2-nonenal. That means 9 carbon. Now, that's really significant because this is a bad player, this 4-HNE it's called, and we're going to take a look at it in a second. That compound can induce a lot of oxidative damage, and in fact, it's been well described as causing oxidative damage in the human cornea. The gene is located within a syndrome that is, we have isolated this gene is defected in the Smith-Magenis syndrome, which is involved in this corneal degradation. And it's been localized, the gene's localized to the human chromosome 17. That gene can be multiply, alternatively spliced, and some of those splice variants are null proteins, so they're not active. Hence, you get a buildup of that 4-HNE from ultraviolet light, and when you have a complete loss of that, you can get this Smith-Magenis syndrome, excuse me. Okay. Now, another very important issue here is a deficiency or a complete ablation of this ALDH3A1, okay, the one we're talking about here, can lead to another disorder, not associated with UV light directly, but it is a subcutaneous skin disorder. It's also called an ichthyosis. Now, ichthy means fish. So this is the very rare autosomal recessive disease known as Jorgen Larsen syndrome. And I hope I'm pronouncing my Scandinavian well enough, but that basically is a disease found in autosomal recessive, very rare disease you find in certain Scandinavian populations. And that causes a scaly skin, a fish scaly skin. It could also cause neurological damage. So it's not at all a benign disease. Okay, so that's the enzyme. Now, because I'm a biochemist, I like to show you what the enzyme does. Okay, so here's your substrate. Your substrate is this aldehyde. And the enzyme actually acts just like a Lewis base. Okay, so that means it's going to be a nucleophilic attack. Because it's going to be an electron donor to these moieties in the enzyme, in the aldehyde dehydrogenase. So you're going to oxidize that aldehyde, and there's your acid. Okay? So the oxidation then is going to be catalyzed by a Lewis base catalysis, okay, for those of you that like that sort of thing. All right. Now, this particular diagram I obtained from a very recent paper, Chem Communications, talking about the actual mechanism of this enzyme. So if you like enzyme mechanisms, I'm not sure if this is probably not open access, but you can, if you have access to a library, you can go in and take a look at this paper. It's actually quite interesting how this particular catalysis occurs. All right. Now, why else do we want to know about alcohol dehydrogenase? We just told you a couple of rare, quite frankly, syndromes, right? Well, also keep in mind that you build up a lot of aldehyde just during normal aging, and of course, as I said, because of UV light, okay? We're all exposed to UV light. Now, let's take the classic example of ethanol ingestion. Remember that ethanol, which is a toxin, it's not recommended to take this in the diet at any level. Ethanol is, first of all, catalyze an oxidation of ethanol to the acid aldehyde carried out by an alcohol dehydrogenase. That acid aldehyde is a bad player. It's cytotoxic. So fortunately, there is an aldehyde dehydrogenase, and that's the enzyme we're talking about. And that aldehyde dehydrogenase is going to take the aldehyde and turn it into acetate. That is acetic acid, the acid. So in normal alcohol metabolism, and in my concerted opinion, any ethanol ingestion is not a good idea. But when a person decides to make that mistake and take in this drug ethanol, alcohol dehydrogenase converts it to acid aldehyde. And then when you have a very active, competent, potent aldehyde dehydrogenase, you make the acetic acid, you remove the cytotoxic compound. Now, what happens when you take in too much ethanol or chronic ingestion of ethanol over time in life, you build up a lot of acid aldehyde. You have maybe a mutation in the aldehyde dehydrogenase, or you get a loss in aldehyde dehydrogenase, or an overloading of that enzyme. You build up acid aldehyde, and this is a bad player. This can cause cytotoxicity. In fact, it's one of the major causes of cirrhosis of the liver. Okay. These next few diagrams here are coming from a paper from Physiological Reviews published some 3 years ago. Okay, so I told you that 4-HNE, that's that 4-hydroxynonenal, shown here as this sort of squiggly line here, is a really bad player. We can make it a lot of different ways. You don't get it from ethanol, but you can get it from making reactive oxygen, for example, exposure to UV light, or just the spontaneous induction of reactive oxygen, for example, from the electron transport chain, right? Okay. So what happens, right? Well, let's take a look here. So this diagram is trying to show you what's written here. So you can look at the diagram. This is supposed to be the nucleus and the mitochondria, and there's the proteasome. So what does it do when you build this aldehyde up and you don't have the aldehyde dehydrogenase, okay? You're limiting it. You have a mutation in it, for example, or you have a decreased amount of expression of it. First of all, it's produced via that reactive oxygen species-mediated lipid peroxidation. This happens all the time. You make a lot of reactive oxygen directly from polyunsaturated fatty acids in the plasma membrane, for example. You also get it in the mitochondrial membrane. Okay, we also know it can happen in the electron transport chain, and even some of this can be generated. So what goes on then? You make 4-HNE. It reduces membrane integrity, so the membrane no longer couples as it's supposed to, so you don't have normal signal transduction pathways, for example, neurotransmission, for example, transport of organic compounds across those membranes. You can also inhibit proteasomal function, so that means you can't turn over proteins. Another aspect of protein metabolism, you trigger the unfolded protein response, the UFR, which is an endoplasmic reticulum mediated SOS type of response. And if you overload the unfolded protein response, you will build up a lot of unfolded proteins, and this is toxic to the cell. Remember, the ER is involved in making proteins that are going through some kind of secretory pathway, or proteins that are destined to get buried into membranes, any membrane, endomembrane in the cell, or the plasma membrane, right? So when you overload the unfolded protein response, it's a bad thing, and 4-HNE is going to do that. It's going to trigger it and overload it. Likewise, it's going to inhibit the electron transport chain because it's going to try to be an aldehyde in that those oxidation reactions, and it's not going to work. So it's going to be an inhibitor. Same way with the dehydrogenases in the Krebs cycle, the TCA cycle. It's going to inhibit those normal dehydrogenases. For example, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, malate dehydrogenase, succate dehydrogenase. It's going to inhibit, because it's going to be a potent inhibitor of the alcohol dehydrogenase itself, because it's not a common substrate. So once it gets in there, it's going to block that. So any of the normal aldehydes that are made, for example, acetaldehyde, is not going to be processed correctly. It's going to cause an increase in mitochondrial membrane permeability and dysfunction. That's not a good thing. That can lead to apoptosis. And it's going to cause DNA damage because it's an aldehyde, and the aldehydes wreak damage on macromolecules such as proteins, DNA, and lipids. Okay, so it's a really bad player, right? And this is all because of reactive oxygen inducing the production of this. That's one thing, but then not having its removal via the aldehyde dehydrogenases. So this is just about the end of the talk today. Yay. Here's a paper that just came out. Well, I guess it's about a year old now, actually. Again, it's talking about our good friend here, the aldehyde dehydrogenase 3A1. And here, these folks have determined that this enzyme actually also plays a functional role in the maintenance of corneal epithelial homeostasis. Now, that's really interesting. Okay, we're back to talking about the cornea. Now, what are we talking about here? Take a look at the enzyme. The protein actually will, in some yet unknown mechanistic way, interact in the cytosol. Remember, this is cytosolic dimer protein with the protein P53. Now, we know that P53 is a tumor suppressor protein, right? The classic tumor suppressor gene product, P53. Well, when wild type, that is non-mutated form, this paper talks about using a mutant form. That's why they show you in the diagram. Again, this is a +1 paper. Okay, so it's open access. You can get this paper and read the whole thing. That enzyme binds to P53, goes into the nucleus. Okay, there it meets up with another protein called this MDM2. MDM2 then allows P53 to be monoubiquitinated, and that sequesters P53 in the nucleus. Now, why is that important? That's where P53 works as a checkpoint inhibitor. So when P53 is in the nucleus, guess what it's going to do? It's going to block cell proliferation because it's going to block DNA replication. That's a good thing, right? So this is a good process. Now, besides that, once the protein dissociates from P53, it also seems to have some role in activating its own suite of transcription factors. So it's going to be involved in gene expression. And it's going to cause those genes which normally are involved in cell proliferation, the mitotic type gene complexes, to shut down. And it's going to enhance terminal differentiation of these epithelial cells, which is a good thing. Go through terminal differentiation. What's that mean? You're not going to go through that mitosis. You don't go through mitosis, you're not going to get proliferation. You don't get proliferation, you might then be able to ablate or decrease tumorigenesis, right? So that's the key here. good player all around, removes that nasty aldehyde, and also here it is functioning as a carrier of P53 to suppress tumors, as also works with its own suite of transcription factors, which further causes terminal differentiation of these corneal epithelial cells, which further then can limit the amount of tumorigenesis. So basically, aldehyde dehydrogenase to remove active aldehydes, and it aids in P53-mediated prevention of, sorry, you can't quite see that, the ultraviolet radiation-induced cell proliferation. So this is really a good player. So we get a lot of ultraviolet radiation. That we want this protein to be active. We want it to be able to carry P53 in the nucleus. We want it to be able to shut down proliferation and cause terminal differentiation. So again, summarizing, aldehyde dehydrogenase, both the 1A and the 3A1 protect interocular tissues from UV radiation, which causes oxidative damage, partially because it produces reactive oxygen species. There's an inverse correlation between the 3A1, the player we've been talking about, right, that dehydrogenase, and corneal cell proliferation. So high amounts of that enzyme, because of this reaction, because it removes those aldehydes, you get a decrease in cell proliferation. See here, boom, decreased. And the dehydrogenase decreases corneal epithelial proliferation through both. This is like the summary sentence, non-enzymatic and, of course, the enzymatic dehydrogenase pathways, okay, making the acid. So thanks a lot for your attention today. Again, I'm Dan Guerra from VerEvMed. There is our email. You can contact us directly with your questions, and we will dig into the literature with you and come up with answers based on verification of the evidence we find in that vast array, that manifest of publications that continues to build every week, every day, indeed. So we would be glad to work with you on your given research project and dig into the literature with you and help verify it and pull it out and examine it and interpret it. There's our website. Please visit our website. It explains a lot of what we do. And remember that we are scientists. We're not medical doctors. We are scientists. So I'm a PhD scientist. My colleague is a PharmD scientist. And what we do together is we verify published evidence in medical biosciences. Thanks a lot. Again, this is Dan Guerra saying, talk to you soon. Bye.